Hello and welcome to this session on angiogenesis. Now, this is one of the other roads to cancer. In the last week, we saw how cell signaling and cell death plays a role in cancer progression, and today we'll look at angiogenesis, or how cancer cells switch on the blood supply. First, what is angiogenesis? Angiogenesis is defined as the formation of new blood vessels from pre-existing vessels. The smallest one is the capillary. So typically it's the formation of new blood vessels from a pre-existing blood vessel, and typically done because of the migration or movement and division of a specialized type of cell called as the endothelial cell, which is found on the inside of a blood vessel. And angiogenesis is not unusual at all. It occurs normally in most physiological events. For example, if you cut yourself, the wound has to heal, and along with that, the blood vessels have to also form. In females, angiogenesis occurs every month in the process of menstruation when the blood vessels have to reform inside the uterus. In tumors, on the other hand, this abnormal angiogenesis results in tumor cells growing, invading, and spreading to different sites. So how does this work? Now, typically, any tumor will grow no bigger than the size of a pea up to a point, because even if the cells try to grow, they don't get the oxygen and nutrients, so growth will be inhibited. The cells will literally be dying. However, after a point, these tumor cells start to secrete factors called as angiogenic factors. And these factors or proteins trigger angiogenesis. They trigger the branching out of the pre-existing blood vessel, which starts to divide and proliferate. And as a result, this can feed the tumor, and the tumor can now start to grow. And this process or triggers or switch from a in situ tumor, the pea-sized tumor, to a growing tumor can vary in different types of tumors. Very short in some aggressive tumors, and much longer in some slower growing tumors. And eventually this tumor can grow so big that it can spread to other organs. How do tumors switch on angiogenesis? Now, this typically is a balancing act. Almost all tissues will have proteins which either trigger angiogenesis or stop angiogenesis. And in most tissues, the overwhelming number or amount of proteins is anti-angiogenic. That's why there is no stimulation of vasculature in most tissues. And so this balancing act between anti-angiogenic or inhibitory proteins or factors versus angiogenic factors is what decides the fate of that cell or that tissue. Don't be worried about the acronyms on the side. I've just written it down to help you understand some of the growth factors, VEGF or vascular endothelial growth factor, etc. We'll encounter these later on. Now, another point to note is that the blood vessels that do form in tumors are abnormal. Unlike normal tissues where the branching network is very systematic, very organized, this is the complete opposite in tumor cells. So this vasculature is haphazard, it's leaky, it's sort of disorganized, and not just in appearance, but also in how it works morphologically and in physiology terms. So these branch networks are tangled, they have holes in them, they're leaky, some sections are very thick, some sections are thin. So all this results in a host of consequences for the tumor. For example, here in this image, what you see here are the cancer cells in green, and what you see in red are endothelial cells. And you can see a cancer cell is inside this blood vessel, showing that this tumor cell has come out and gone into the blood vessel. And this is also shown in the schematic on the right, which shows the pores or the holes inside the blood vessels. And this electron micrograph indicates precisely that, where you can see with the arrows the size of the holes. And what this results in is serious consequences to the cell. It can make a bad situation worse. So the tumors can become very hypoxic, they can become very acidic, and as a result of all this, it increases the chances that these cancer cells will move from one location to another or spread. And this can result in several other things as well. For example, as I mentioned before, some sections are thick, some are thin, and some sections of the tumor might have a lot of blood vessels, some might have very little blood vessels. And as a result, the drugs that are given, chemotherapeutic drugs or radiation, for instance, doesn't actually work on all the tumor cells equally. And this results in resistance to the drugs because this drug is sitting there for a long time and not really doing anything much. It also, like I said before, it increases the chances of metastasis or spread of cancer cells from one location to another. So let's look at how the molecular pathways work. Now, before I go into that, I just need to tell you that there will be a lot of acronyms, and let's go through the pathways a bit slowly. So if you familiarize yourself with this image, first of all, what you see on the top are the cancer cells, and what you see below, the cancer cells are in green, and what you see below is a cartoon of a blood vessel in red, and you have the endothelial cells lining this blood vessel. And what you see on the left-hand side is simply the list of some of the proteins we'll encounter today. So starting off with tumors. So what tumors do is they secrete this factor called as the vascular endothelial growth factor, VEGF. VEGF goes and binds to a receptor, a protein that is stuck on the surface of the endothelial cell. And when this receptor is activated by this binding, it leads to several things. First of all, it activates the endothelial cells. These endothelial cells start to secrete several things, and these endothelial cells start to secrete other factors. The activation of the endothelial cell can also lead to the activation of proteases. Proteases are enzymes that digest protein. So these soluble proteases are activated and they start to chew up the surrounding protein, making a physical space where the cancer cells can move into. Activation of the endothelial cells by VEGF binding also leads to activation of another receptor called the integrins, shown in the green triangles here. Now, integrins are like Velcro. They help cells to move physically. So these integrins enable the physical migration or movement of these blood vessels or endothelial cells towards the tumor. So that's the source of VEGF. So the endothelial cells start to go or branch out to the tumor cells. There are other proteins that are also activated. An example is the PDGF, platelet-derived growth factor, which again activates other receptors on the stroma or supporting cell. And this lends itself very well to designing drugs that target angiogenic factors. Now, there are several anti-angiogenic drug targets. One example here is Avastin, which inactivates this VEGF itself, and it has undergone clinical trials quite successfully for certain types of brain cancers called as glioblastoma, recurrent glioblastomas, but it wasn't very good for colon cancers. Sutent is another drug that actually blocks this receptor for VEGF, so it prevents the activation of endothelial cells. There are pros and cons, and there are some good success stories with using anti-angiogenics in clinical trials. So let's go over the key concepts again, as you can see on the screen. And next time we shall look at the other pathways to cancer progression.